All right, so you saw that we have lungs, we breathe through the bronchus, and you have all these tubes that are branching, and then it ends up in an alveoli. So the air ends up in an alveoli. And why is that? Why do we have these structures? Why do we have all these structures? Why does the air have to end up in a tiny sac like that? Because ultimately air is exchanged with a blood vessel via diffusion. And diffusion, you can only transport molecules a small distance in a small amount of time. Okay? So that's why you have these complex structures. So oxygen can diffuse to the blood, and carbon dioxide can diffuse from the blood to the alveoli. Okay? So you can diffusion transport stuff only short distances. Okay. So that's why we have these structures. So here's alveoli, and here's air in the alveoli, and oxygen diffuses from here to the blood. This is the capillary, and we learned that in a capillary, see the red blood cells go single file. Okay. And we'll talk more detail about that. Oxygen diffuses from here to the blood vessels, and carbon dioxide diffuses from the blood to the alveoli. Okay? And for diffusion to happen, oxygen needs to be at a higher concentration here. Okay? And CO2. So oxygen needs to be at a higher concentration to diffuse this way. And if you want carbon dioxide to diffuse that way, carbon dioxide needs to be at a higher concentration in the blood. Okay? And so we'll talk about some of the numbers. Okay? So diffusion is happening across the alveolar walls and the capillary walls. Okay? And so these walls are thin enough and they're permeable to these gases for diffusion to occur. Okay? So oxygen diffuses from the alveoli to the red blood cells, and carbon dioxide diffuses from the blood to the alveoli. Okay. So there's actually 500 million alveoli, like you saw before, and they're each about 200 micrometer in size. Okay, and the size of a single cell is about a micrometer. So the alveoli are about 200 micrometers big. Okay. And the cell wall is about 0.4 micrometer thick. Okay, so that's the dimensions of that. Okay. So blood passing through capillaries adjacent to alveoli exchange blood passing through exchange gases with air via diffusion. Okay, so that's the process. The last mile, ultimate mode of exchange is diffusion. Okay? So diffusion proceeds in the direction from high to low concentration. Okay? And the rate of diffusion will depend on the concentration difference. Okay, so if you increase the concentration of oxygen here, more oxygen will diffuse. Okay, so the rate of diffusion depends on the concentration. Okay? So actual rate of exchange is greatly affected by the presence of hemoglobin in the blood. Okay? So in addition, your body has optimized oxygen transfer. Okay? The presence of hemoglobin binds to oxygen, and so it aids the rate of diffusion of oxygen is aided by the presence of hemoglobin. So when oxygen binds to hemoglobin, it behaves the concentration of oxygen here behaves like it's lower. So that means the concentration difference is higher and more oxygen diffuses. Okay? So the actual rate of exchange is affected by the presence of hemoglobin in the blood. Hemoglobin has a chemical affinity for oxygen, and its presence allows the blood to carry far more oxygen than it otherwise could. Okay? So the presence of hemoglobin helps that. The exchange also depends on blood acidity, presence of carbon dioxide, and temperature. All these dependencies are cleverly designed to enhance oxygen transfer. Okay? So your body is very finely tuned to enhance oxygen exchange. All right, here are some numbers. Okay. By the way, what's the pressure in this room? What's the composition of atmosphere? Okay, so it's 79% nitrogen, about 20% oxygen, and 1% other stuff, okay? Carbon dioxide and all that stuff. So that's the composition. And what is the atmospheric pressure? It's about 15 psi. Okay, so what this atmosphere comes is, this is atmospheric pressure. That means air is exerting a force on every square inch of 15 pounds. It's exerting a force of 15 pounds. Okay, what the partial pressure means is this 80% of that pressure is coming from nitrogen molecules, and 20% of that pressure is coming from oxygen molecules. So the pressure exerted by oxygen molecules is 20% of that, 10% is 1.5 psi, 20% is 3 psi. So the pressure exerted by oxygen molecules is 3 psi, and the pressure exerted by nitrogen molecules is 12 psi. That's the partial pressure, okay? All right, so it's proportional to the amount of molecules you have. Okay, so about 20% of the atmospheric molecules are oxygen molecules. The partial pressure of oxygen is 20%. Okay, so if we do it in millimeters of mercury, atmospheric pressure is 750 millimeters of mercury, 760. 20% of that would be 75 plus 75, 150 millimeters of mercury. So the partial pressure of oxygen in the atmosphere is 150 millimeters of mercury. Okay? So this is expressed as partial pressure of oxygen is 150 millimeters of mercury. All right. Now the partial pressure of oxygen in the lungs is only 105 millimeters of mercury. Remember, what we're trying to do is we're trying to figure out, in the alveoli, oxygen goes from here to the blood, and we are concerned with the oxygen concentration of oxygen in the alveoli. Okay, and here we go. Here this is saying that the partial pressure of oxygen in the alveoli is about 30% less than in the atmosphere. So what's going on? Okay, we want a higher concentration in the alveoli to exchange. What goes on, and see, every time you breathe, you're not replacing all the air in your alveoli. You're replacing only about 14% of the air in the alveoli. Okay, so there's stale air there. So the steady state concentration of oxygen is only 105 millimeters of mercury of oxygen. Okay? That's the partial pressure. Now, by the way, so when they tell you to take a deep breath, when you take a deep breath, you can replace from 50 to 100% of, you can refresh the air in your alveoli. Okay? And so increase the concentration of oxygen in your alveoli. Okay. Air in the lungs has a partial pressure of 105 millimeters of mercury, and in blood it's only 40 millimeters of mercury. So concentration of oxygen in the alveoli is greater than in the blood. So oxygen from alveoli will diffuse to the blood, and vice versa for carbon dioxide. Carbon dioxide concentration in the blood is higher, and it diffuses the other way. So we haven't given you the numbers for concentration. Now, partial pressure is being used as proxy for concentration. Okay. So since the concentration of oxygen in the lungs is much greater than in blood, oxygen will transfer into the blood. Okay. And vice versa for carbon dioxide. Yep.
Shallow breaths versus deep breaths.
Mm-hmm.
Long
Yeah, this is a normal breathing. You're only replacing about 14%.
Is that part of what triggered the yawn?
Uh,
To get more oxygen sort of thing?
I don't know what. So part of the deal is the reason we do that is you don't want the concentration to fluctuate too much. So that's what it's designed for. Okay. I don't know if you were here. Do you remember the numbers for, anybody remember the numbers for a manatee? So a manatee replaces 90% of the air in its lungs all the time. So we're doing only 10 to 15%. So what's the deal with that? So that's why manatees can stay underwater 20 minutes with one breath. Okay. Why do they have to do that? Do you guys remember the fat level of manatees? 7%, that of an elite athlete. Manatees eat plants. That's not high food concentration. You got to be eating all the time. It's like an elephant. It is an elephant in the water. So you got to be underwater. Everything is optimized. There is a good reason for everything. You just got to ask the right questions. Okay. All right. The next topic we'll talk about is wetting. Okay. So we'll learn a few things. One of the things we'll learn is how do tall trees like redwood trees get water up to the leaves. The leaves are about 100 meters above the soil level. 100 meters is 1.5 football field. Well, a little. So from goalpost to goalpost. How do you get water up there? Okay. So a drop will bead up into an almost perfect spear on a waxy surface. So on a waxy surface, the water will bead up into a spear. On a non-waxy surface in glass, see, it spreads out like this. So what's going on? All right, so you want to understand two terms, cohesive forces and adhesive forces. Okay, so this is a water molecule, here's a water molecule. I'm just, so these are supposed to be neighbors. The forces between them, this is cohesive forces. Okay? This is a water molecule, and this is the surface molecule on which it is. The force between them is an adhesive force. Okay, so those are the two forces. So forces between like molecules are cohesive forces, and forces between the liquid and the surface molecules are adhesive forces. Okay. So if a liquid spreads out like this, that means the adhesive forces are greater than the cohesive forces. Okay, so for this guy, adhesive forces are greater than cohesive forces. For this to happen, cohesive forces are greater than adhesive forces. Okay? So when you wax your car and the water beads up into droplets, what have you done? And you don't want the water to wet your surface and then get dirt on the car because there's dirt mixed in the water. When you wax your car, what you have done is you have reduced the adhesive force, the force between the water molecules and the car surface. Okay. All right, so keep that in mind because the next few slides depend on this idea. Okay. So when a water drop beads up into an almost perfect spear on a waxy surface, then the cohesive forces are greater than the adhesive forces. Okay. So the water molecules do not attract as strongly to the waxy surface. The same water drop would spread out into a flat puddle on a clean glass surface. On a clean glass surface, water molecules attract the glass surface stronger than between themselves. Okay? Adhesive forces are stronger. So we define this contact angle. So if the contact angle is less than 90 degrees, then adhesive forces are greater than cohesive forces. If the contact angle is greater than 90 degrees, then the cohesive forces are greater than adhesive forces. Okay? So this contact angle tells you which situation you're dealing with. Okay? If the adhesive forces that pull liquid molecules towards the surface are larger than cohesive forces, if adhesive forces are larger, then the contact angle is smaller than 90 degrees. Okay? So adhesive forces greater than cohesive forces, and cohesive forces greater than adhesive forces. Okay. All right. Wettability is an important parameter, so we want to know what surfaces are wettable. A surface is said to be wettable or hydrophilic. Adding a surfactant causes the drop to spread even more. So let's say you have water on a waxy surface and you add soap to the water, it'll spread out like that. What a surfactant like soap does is it reduces the cohesive force. Okay? And when water wets a surface, that's when you can wash away stuff. You want the water molecules to, bind to dirt and all that. So wettability is important. It's said to be wettable or hydrophilic. Adding a surfactant causes the drop to spread even more. Better wetting the surface with a smaller contact angle. Okay, wettability is an important parameter in interaction of biological fluids with surfaces. Okay? So it's whether this is that is blood serum interacting with a microscope slide or saliva interacting with your dental enamel. Okay? All right, so this is called a wettable surface. A surface is wettable if the adhesive forces are greater than cohesive forces, and this is a non-wettable surface when the adhesive forces are less than the cohesive forces. Okay? So this is a non-wettable surface. So waxy leaf surface is very hydrophobic, as are the surfaces of non-stick cookware. So non-stick cookware are, all you have to do is make sure you put a chemical on the surface of the cookware which is not attracted to water molecules. That's a non. Okay. Or oil and stuff like that. Okay, so again, we're talking about this contact angle. If the contact angle is greater than 90 degrees, then cohesive forces are greater than adhesive forces. Okay, so here's contact angles for various materials. The forces between mercury molecules in mercury is the cohesive forces are very strong. So mercury and glass, you see the contact angle is 130. That means cohesive forces are strong. Water and glass is about 30 degrees. So water molecules and glass, the strong force between water molecules and glass molecules is stronger than force between water and water itself. Okay, so when contact angle is less than 90 degrees, blood and glass 25 degrees, water and clean glass, it's almost 0 degrees. Okay, if you have a very perfectly clean glass, water would spread out. Okay, it'll wet the whole surface. Okay. Water spreads into a thin sheet on very clean, very smooth glass. Okay. By the way, contact angles are very sensitive to surface roughness and cleanliness. All but the cleanest surface have trace amounts of other molecules in them, and that's why you have this 30-degree contact angle. If you had perfectly clean glass, water would spread out to a molecular level thinness. Okay, but you need a perfect glass. In more typical cases, such as raindrop on your windshield, water on glass forms flattened drops. Okay. So your windshield always has imperfections and other molecules on it. Okay. All right, here's why this is important. Okay, so this is when the adhesive forces are greater than the cohesive forces. So what is happening here is, okay, this is the water molecule attracting to the glass molecules. When these forces are strong, see, the meniscus is like that. Okay, so the molecules of the glass are pulling up the water. When the adhesive force between mercury and glass is weaker than the force between mercury molecules, you form a meniscus like this, and this liquid will go down. You have a meniscus that forms like that. All right, so this is, let me write it here. This is when adhesive forces are greater than cohesive forces. And you have this meniscus when cohesive forces are greater than adhesive forces. All right, well, why were we interested in this fact? Okay, so if you take small tubes, small diameter tubes, water, because of these adhesive forces, water will rise up. So the attractive force between the glass and the water molecules is pulling this water up. For mercury, those forces are pulling the mercury down. Okay? The force between the glass and the mercury is pulling it down. Okay? So this is known as the capillary effect. The narrower the tube, the higher the liquid will rise. Okay? And so the narrower the capillary, the greater the fluid will rise. Okay. Now, this formula gives you the height to which capillary will rise. The height depends on the surface tension, okay, and cosine of the contact angle. Okay. So if the contact angle is an acute angle, this is positive, and the liquid will rise. Okay. And you see the height to which it rises is inversely proportional to the radius of the tube. So smaller the radius, smaller the capillary tube, higher the water will rise. Okay. It also depends on gravity. So the same tube on the moon where the gravity is less would rise higher. Okay. And we use this capillary effect in many situations in our body. Okay. So if you have a thin tube, it'll pull up the liquid as long as the adhesive forces are greater than the cohesive forces. Okay. For a mercury, it depresses. For a mercury, the contact angle is greater than 90 degrees, and cosine theta would be negative, and so H is negative, and the liquid falls down. Okay. So if the contact angle is greater than 90, H is negative. All right, so now let's look at some applications. So in a thin tube, water would rise up. Okay? And and what is pulling up the water? It's the adhesive forces. The forces between glass and water molecules, that's pulling the water up. Okay? So that's what's pulling the water up. And here also adhesive forces are pulling the mercury down. Okay? So let's look at an application. Okay, again, you have a thin capillary, and the adhesive forces will pull the liquid up the tube. Okay. So here's an application: a capillary tube used to sample a blood from a finger prick. So you want to sample your blood for, let's say, for diabetic patients. You prick your finger, and then a capillary tube will pull up that blood. Okay, you don't have to have a pump to pull it up. Okay. So a capillary tube used to sample blood from a finger prick has an inner diameter of 0.8 millimeters. So that's the capillary tube. That's the size of the capillary tube. to what height will the blood rise in a vertical capillary tube? So to what height will the blood rise? There is the formula. There's the surface tension. So there's the surface tension of blood. There's the contact angle, okay? Density of blood, g, the acceleration due to gravity, and there's the radius of the tube. Okay, again, you don't have to know all that calculation, but the blood would rise up to 23 millimeters, 2.3 centimeters, which is about an inch. So blood would rise up to an inch in a capillary in that. Okay, so you can do a pinprick and hold that machine there, and blood will rise to an inch. So about an inch. Blood draws are able to fill a 80 millimeter, that's about more than 3 inches long tube, because the tube is, if you hold the tube horizontally, if you hold the tube, instead of holding it vertically, if you hold it horizontally, you can draw blood up to 3 inches from the capillary effect. And what you're essentially doing is making this smaller by holding it horizontal. All right, so now you understand, if you want to draw blood, you have a pinprick and hold a capillary horizontal, and you can draw up to 3 inches of blood, okay, just from the capillary effect. And what is the capillary effect? The adhesive forces are pulling the liquid up. Okay, adhesive forces are the forces between the glass molecule and the liquid molecule. Okay, here's a more, so the adhesive forces are pulling this liquid up. Here's the example. That's how wax rises in the wick. Okay, so when a candle burns, it's burning wax. So you're running out of wax. How are you getting a constant supply of wax? This heat is melting wax, and that is rising via the capillary effect. Okay? So capillary action plays an important role in many. Wicking is such as movement of water through a paper towel. That's a capillary effect. So in your paper towel, there are small capillary tubes. Okay? Through a paper towel, or molten wax through a candle wick is capillary action through small diameter pores. Similarly, pores through soil help water bring up from the deeper layers to the root zone of plants. So water coming up in soil is also a capillary action. Okay? Again, you guys understand that capillary action is due to adhesive forces between the walls of this tube and the liquid that's pulling the liquid up. Here's another place where your capillary action is used by your blood. So here are tear glands. Tear is produced to moisten your cornea, clean your cornea, and another important thing, it forms a smooth optical surface. A layer of water will form a smooth surface for the lens, and so you can form a sharp image on your retina. Okay, so tears do many functions. Its main function is, the function of the tears is to lubricate, moisturize, protects the eye surface, and also provides a smooth optical surface. You want a smooth optical surface to make a sharp image on your retina. Okay, so you're constantly producing tears. So now, if you're constantly producing tears, what do you have to do? You have to constantly remove the tears. And these are capillaries, they'll pull the tears out. Okay, and then when you blink, what happens is you squeeze these things and release them, so you're making partial vacuum, and that sucks the tears in. So you're using capillaries to draw those tears, and then using partial vacuum, you're sucking them out. And then your tears get deposited in your nostrils. Okay. The punctum, this is the punctum, are about 0.2 to 0.3 millimeters. Okay, just like, see, that's 0.8 millimeters. Okay, so remember, for the capillary action, smaller the tube, higher the tube is, more of the water is sucked up. Okay. Capillary action helps tear and enter the puncta, and every time you blink, tear are pumped into the cannulicus. Tears are pumped there and then eventually drain in the tear duct. So we use capillary actions everywhere. Yep. Now we'll figure out how does water rise to into the leaves of these tall trees. Like I said, these trees are taller than a football field. Okay? Now capillary action is what brings up water in small plants. But capillary action cannot lift water till more than about a meter. A meter is the height of this desk. So capillary action can only lift water up to, at best, a meter. So now we'll have to figure out how to bring water to the leaves of tall trees like this. Okay, so capillary action, again, was a result of these adhesive forces. Okay, so what brings up water is essentially the following. Okay. It's these water molecules attracting each other. So when you pull this water molecule, that is attracting this water molecule. It's like a chain link that you're pulling up. So that's how you're lifting the water all the way to 100 meters. So it's the force of attraction between water molecules, the cohesive forces that's responsible. Okay. So when this water molecule evaporates from the leaf, it pulls on this molecule, which pulls on this molecule, and that's how this thing is rising. What would happen if you cut this thing? What would happen if you put an air bubble in there? If you put an air bubble in there, now you've broken up the chain link and you can't raise the water anymore. So remember that, and we'll mention an important point, okay? Okay, so capillary effect cannot raise water more than a meter, okay? So what we are trying to answer is water can ascend more than 100 meters in the tallest trees, and we want to know how. So because of cohesion, water and other liquids can, under the right condition, be placed under tension. Okay, what that means is, let's say you have a wire or a rubber band, you can pull on this rubber band. You can apply a force on the rubber band and stretch it. So what we are saying is, you can take a column of water and you can place it under tension. This won't break. That's because of the attractive forces between the water molecules. And you'll see the numbers, they're pretty amazing, okay? Because of cohesion, water and other liquids under the right condition can be placed in tension like a rope. Water gets to the leaves not by being pushed from below, but by being pulled from above. Okay. So, and this is due to the strong hydrogen bond between water molecules, the strong cohesive forces. Okay. In a large tree, several liters of water evaporate per minute. So, excuse me, hello there, can you hold up that's about a liter. So several liters of that evaporate from a tree every minute. Okay. Water evaporation takes place through stomata on the outside of the water molecules exert attractive forces on each other. Molecules leaving the pores pull and tug on the water molecule that remain in the leaves. So a water molecule evaporating is pulling on the rest of the molecules and pulling the water column on. Okay. Those in turn pull on molecules in the xylem and so on all the way down to the roots. Okay. As a result, the entire water column is under tension, just as pulling one end of the rope places the entire rope under tension. Okay. So here's a water column. You're pulling on this water, and this water column is under tension. So the question is, what kind of tension can it bear? Okay, so you're lifting this water column up like that. It's like you're pulling on this top link of the chain, and all these interconnected links are being pulled up. The stress for a 100-meter tall tree is easily calculated. Stress is tension divided by the area of cross-section. The tensile stress needed to support a 100-meter column, so the tensile stress that you need is 1 megapascals, 10 atmospheres. It's force per unit area, okay?
147 pounds,
What's that?
147 PSI.
Yeah. So what tensile stress will break it? It can bear up to 30 megapascals. So that's what will break it. Okay? So it's amazing. Okay? So to support, all you need is 1 megapascal, okay, to pull it up a 100-meter tree. The water column is not just hanging in equilibrium, but is moving upward through the xylem. Forces are needed to keep a viscous fluid moving through a pipe. For trees, the additional force needed to move so you're pulling up this water column, but there's a friction with the xylem, so you need additional force to pull it up, okay? For trees, the additional force needed to pull is the same as the force needed to support the weight of the fluid. So you actually need, so because of the friction, you need an additional force, and so you need 2 megapascals to pull up this water up the tree. Okay? The tension due to the cohesive forces in the water actively pulls up the water up the tree. Okay? Yeah. So can water do this without the water column breaking? So now here's an important fact. Can water do this without the water column breaking? If the water column breaks, then the tension all goes away and the water is no longer rising. Okay. Yeah. But very narrow tubes of water supported by a hydrophilic surface of the xylem can withstand a surprising amount of tension. Okay, so these water molecules are also attracted to the xylem surface, and the water molecules are attracting to themselves. So you can support a big amount of tension there. Tensile strength is what tension can you apply before this column would break, and that's 30 megapascal. And to lift water up, all you need is 2 megapascals. Okay, so you're far away from breaking this column. Okay, in fact, potentially that tells you that you could have a tree that's 15 times taller than a redwood tree. Okay, but then you would be living on the edge. Okay, so this tensile strength is nothing that like a metal, but it's adequate to lift water up to the tallest trees. Okay. Okay, 2 more features are worth noting. First, the entire system is powered by the sun. Okay, remember, how did the water molecule evaporate? It was using energy from the sun. So you don't have any motor pulling this up. You're using this energy of the sun. Okay. So no expenditure of metabolic energy is needed. None of your ATP molecules are needed to pump up the water, which is good. Okay, you're using energy of the sun. Second, bubbles are the enemy of pulling liquid under action. So if you have a bubble forming, then you're cutting off the supply of water. All right, a bubble cavity or embolism breaks the water column and stops the flow. Okay, just as effectively as cutting a rope. The narrow diameter of the xylem vessel may be an evolutionary response to this danger. Because bubbles don't easily form in narrow tubes. So the xylems are narrow because bubbles don't form in narrow tubes easily. Okay? But freezing is another story. Freeze-thaw cycles tend to introduce bubbles that prevent the xylem vessels from reactivating in the spring. So if you have a freeze and then you thaw, then you introduce bubbles. So how do you fix that problem? You grow new xylem every spring. Okay? So trees in climates with cold winters grow a new set of xylem vessels each spring under the bark. These become the growth rings. So those are the growth rings. Yep. I'm not sure. So you could ask the question to trees growing in the equator. There you never get, well, maybe at the heights, you can, in Kilimanjaro at the equator, there's snow on the peak. Okay. So anyways, so that's very interesting. Okay. All right. So after this, we have 2 more lectures. We don't have a class next Tuesday because my Thursday class is canceled. Right, next week is Thanksgiving. Is next week Thanksgiving? So we don't have a class next Tuesday. Okay. And then we have 2 more lectures, and I have to get up to slide 150. We have 150 slides. Okay. So now we'll look at fluids in motion. Okay. My goal was to get to slide 105 today. We'll see how far we get. Okay. Now we're looking at fluids in motion. Okay. So what's happening here? What is this? Smoke. So what happens is here's a, what is this called? What kind of stick is this? Incense stick. Thank you. So here's an incense stick that is burning. The hot tip, what does it do? It makes the smoke expand, the air expand. The air gets lighter than the surrounding air, and Mr. Bernoulli tells us if the density is less than the surrounding fluid density, this smoke will rise. There's a buoyant force on it, so this smoke is accelerating, and it's getting faster and faster, and once it reaches a critical speed, see, it's laminar till now, and once it reaches a critical speed, it becomes turbulent. Okay, so a liquid going faster than a certain critical speed becomes turbulent. Well, why do we care about this? You don't want turbulent blood flow in your blood vessels. If flow becomes turbulent, to maintain a flow rate, you have to have a push harder. Your heart would have to work harder. So you don't want turbulent flow at all. When a doctor is using a stethoscope, that's what they're looking for. They're looking to hear if there's turbulent flow. And soon as there is turbulent flow, that's a sign of trouble. Okay? So what can happen? So you see, if you have a blood vessel, if you have plaque deposits, we'll see when the tube gets narrower, blood flows faster. And if the blood is flowing faster than a critical speed, this flow becomes turbulent. And like I said, that's a sign of trouble. And this guy is the trouble. Okay? So that's the sort of stuff we're interested in. We'll assume, so we'll talk about various laws. We'll consider fluids in motion, blood flowing through your veins or air moving through your in and out of your lungs. So those are fluids. Fluid flow is a complex subject. Many aspects of flow, especially turbulence and formation of eddies, are still not well understood. So we know turbulence is bad news, but it's not well understood. Okay. So we'll apply all our laws to non-turbulent flow. All our laws will apply to non-turbulent flow, not to turbulent flow. Okay. Air moving at speeds less than 220 miles can be considered incompressible and all that stuff. All right. We consider steady flow. We are considering steady flow like this. Also, this is steady flow. steady flow where the speed is not changing with time. Here is steady flow, okay? So you can measure the velocity of smoke. So here's a wind tunnel blowing this air, and this velocity is not changing with time, okay? So we'll consider steady flow like this. Yeah. The motion of blood in your circulatory system is always laminar. This is laminar flow. It's flowing in layers, okay? If things are working as they should. Turbulence spells trouble. This spells trouble. This flow is indicative of these plaque deposits. Okay? Turbulence in blood vessels make a very particular sound in a stethoscope, which allows a physician listening to your heart and lungs to detect potentially serious problems in a routine physical exam. Okay, so soon as there is starting, when they use a stethoscope, that's what they're looking for, okay, to see if there is any turbulence. Okay, now I'm sort of going to give you the punchline on some of the slides. The equation of continuity. Okay, so let's say you have a pipe whose cross section is changing. Okay, as long as there is no leaks, here is a simple fact. If 2 gallons a minute are flowing in, how much is flowing out? 2 gallons a minute. Okay, if there is no leak, whatever is flowing in is flowing out. Okay, so this is flow rate. That tells you what volume is flowing in. Okay, so what this is saying is, at this location, A1 is the area of cross section of the pipe, and V1 is the area of V1 is the speed of the liquid. Okay, so A1 times V1 is the flow rate. So if the area decreases, and that's constant everywhere, so if the area of cross-section decreases, this blood flow speeds up. Okay? So that's all it's saying, the equation of continuity. And this fact is used by your body. So your body uses this fact. So for instance, when blood first starts out in the aorta, when your heart pumps blood is flowing at 30 centimeters a second, a foot a second. Okay. If blood flew by that fast near your cells, it wouldn't be able to exchange oxygen and nutrients with the cells. That's too fast for diffusion to happen. So how do you slow down the blood? You just learned that. So here's the secret. So here's the speed of the blood. You want to slow it down. You increase the areas tremendously. Okay? So when the blood flows by near the cell, the blood is flowing at about 0.3 centimeters, 0.03 centimeters a second. So it's slowed down by a factor of 1,000. What does that mean? You have to increase the area by a factor of 1,000. How do you increase the area by a factor of 1,000? You guys understanding what I'm saying? Okay, so here's what you do. You're smart. You break that aorta into 10 billion capillaries. So that's why you have all those capillaries. Okay, so the area of cross-section of all those capillaries is 1,000 times bigger than this area of cross-section of the aorta. Okay, so that's how you slow down the fluid. So now you can exchange oxygen and nutrients with your cells via diffusion. Okay? So your body uses this equation of continuity. Your body knows all kinds of physics. Okay? Flow is faster in narrower parts of the tube and slower in wider parts of the tube. Okay? You see this in your kitchen all the time. There's water coming out of your faucet, and as it falls, as something falls, it's getting faster, so the area of cross section of the tube has to get narrower because the flow rate is maintained. Okay, so you see that all the time. Okay, so the flow rate. So the flow rate is the velocity of the liquid times the area of cross section of the liquid. Okay. In the picture, water speeds up as it falls, and so the area of cross section decreases. All right, what this problem shows you is the following, so you just have to remember the numbers. Okay, so here's the tube through which you breathe. What is it called? It's the average, the trachea. So that's called the trachea or the windpipe. So you have one trachea, and then that branches into two bronchioles. So these tubes go into the lungs. Okay, so these are the first branches. Okay, so there's a flow rate here, air flowing in, and so the flow rate gets divided here. And so what this problem tells you is, given these numbers, they can calculate the diameter of these bronchioles. Okay, and so that's what this problem does. Okay. I'm just going to show you the answers. Okay, and if you're interested, you can look it up. Okay, so an average adult male breathes 12 times per minute. So you're taking 12 breaths a minute, and a total of 8 liters of air. Okay, 8 liters. So again, that glass that you saw, that's a liter, and you're breathing 8 of those every minute. Okay. So the flow rate is, so this is converted into meter cube, okay, per second. That's about how much air is coming in. And by the way, the breathing cycle is not symmetrical. 40% of the time is spent inhalation and 60% in exhalation. Okay. The flow is through a cross-section area, so the speed of air in the trachea, so this part shows you the speed of air in the trachea, 1.3 meters per second. Okay, that's the speed in the trachea. That's what that. So, and you have two bronchi. Okay, so the total amount of airflow through the two bronchi has to be the same. Okay, so twice the speed in the bronchioles times the area of cross section. So the speed pretty much remains the same in the bronchioles, and so now you can calculate the area of the bronchioles. And so it turns out you calculate the area of the bronchioles. So this area plus this area has to equal this area. That's the punchline. And so you can calculate the radius of the bronchioles, and that's ½ an inch. Okay, so the radius of this bronchiole is ½ an inch. That's your calculation, and that turns out to be true. Okay, so that's what that problem shows. Okay, so here's a streamline. Okay, so this is in a wind tunnel, and wind is blowing this way, and you introduce smoke to visualize these streamlines. Okay. So each smoke trail represents a streamline in the fluid. Okay. So now we'll introduce Bernoulli's equation. This is Bernoulli's equation, and this is for ideal fluid dynamics. For an ideal fluid, meaning ideal fluid is something that doesn't have friction. It's not viscous. So this won't apply to honey, for instance. Okay, but most air and water have very low viscosity, so we can apply these to those fluids. Okay. So what this equation is telling you is this term is pressure, this term is the velocity of the fluid, and that is the density of the fluid. Okay, so it's saying pressure at a location plus this term. This is the kinetic energy per unit volume. Again, you don't have to remember all of that. Here's So pressure plus kinetic energy per unit volume, the sum of those two terms is constant at every location, okay? So you take the sum of those two terms, that's constant. So you just saw that if a tube gets narrower, the liquid gets faster. If the liquid is getting faster at any location, that means the pressure is lower at that location. So if this term gets higher, this term has to get lower because the sum of those two terms has to be constant. All right, so as the liquid is getting faster, the pressure is dropping, okay? And that's how planes also fly. So here's a cross section of the wing. This is an airfoil, and it's designed so that, see, this air has to travel faster because it's got to go greater distance than this air. As the plane is flying this way, air is flowing that way. This air is going faster, so the pressure at the top is lower and the pressure at the bottom is high, and that's what keeps the plane up in the air. Okay. All right, so this is Bernoulli's equation. The pressure plus this kinetic energy per unit volume at any location, the sum of those two terms has to be constant, okay? So what I want you to remember is the faster fluid is flowing, the lower the pressure, okay? So pressure decreases. The faster fluid is moving at a point in a streamline, lower the pressure, okay? So here's nature using it, okay? So you see, so if you have a mound like this, air has to flow faster over here, and the pressure is lower here. Okay? So these prairie dogs use this fact. Okay, they got built-in air conditioning. So here's a lower mound, and this is a softer mound. Here's a higher mound, and this is a sharper mound. So air has to flow faster here than here. Pressure here is lower. Pressure here is lower, and pressure here is higher. Okay, in fact, this can be a flat hole. Well, you have a slight mounding because you don't want water flowing in. Okay. And the pressure is higher, and this keeps a constant airflow. Free air conditioning. Okay, so all you have to design is make sure that this mound is higher and it's a sharper mound. You had to build a mound here because you didn't want rainwater to flow in. Okay. And so this pressure keeps the air flowing. Okay. So atherosclerosis, so here's plaque building up in your blood vessels. Okay, let me tell you why it builds up, and then we'll come back to that. Okay, so if there was no viscosity in a fluid, all fluid will flow at the same velocity. If there is viscosity, meaning friction in the fluid, this fluid is flowing the fastest, and the fluid at the surface is almost at rest. So when blood flows in your blood vessels, this blood is, the blood in contact with the blood vessel is almost at rest. So anything in the blood can form deposits on the wall because that fluid is not moving. That's how plaque builds up in your blood vessels. Okay? For blood moving through arteries, the fact that the fluid at the outer edges lingers and barely moves allows deposits to build up on the inside walls. All right, so now let's see. So that's how deposits build up. So here's plaque building up on the walls. But you just saw that the narrower the tube, the faster the fluid is flowing. And when fluid is flowing faster, what happens to the pressure? The pressure drops. The pressure drops, and so your blood vessel is like an elastic. The pressure is what's holding it up. If the pressure drops, it collapses further. The problem gets worse. The blood flows faster. Okay. So plaque is formation on the walls of the arteries, and the disease of atherosclerosis causes a drop in blood pressure at the site of the narrowing. Bernoulli's equation can be used to compute the pressure drop. In severe cases, the pressure loss allows the walls to contract, which squeezes the arteries even more. Okay, so you're making the problem worse and worse. And so that's what the stent does. It'll open up the blood vessel. Okay, and okay, so at least you could have gone that way. All right. So here is you might have seen. Okay, so here's a pipe. Okay, if it narrows, so let's say the outside is not showing you the pipe. Okay, so let's say the pipe narrows. Okay, when it narrows, the fluid is flowing faster, the pressure is lower, and you see the liquid is not rising. Here the pipe is wide, the fluid is flowing slowly, and the pressure is high, and this water is being pushed. So this is a pressure gauge. Okay, it's a proxy to measure the speed of the fluid inside the pipe. Yeah. So lower the pressure, higher the speed of the liquid in the pipe. Okay, so in this thing, velocity is greatest at 0.2. Velocity is the slowest at 0.3, where the pressure is the highest. Okay, so this is sort of a velocity measuring device for the liquid. Okay, so we'll talk about this aortic stenosis, and then we'll stop. Okay, today. So your heart is several pumps. Okay, so blood from the body comes into this atrium. So that's deoxygenated, oxygenated blood has come here. That gets sent here, and this pump sends the blood to the lungs to get oxygenated, which comes back here, then goes to the ventricles, and from the ventricles, the oxygenated blood is sent to the whole body. Okay, so it's a series of pumps, and that's a continuous process. All right, so when you're pumping, the ventricle pumps the blood through the aorta. Okay, so here's the valve. So here is this end-on view of the valve. So this is the valve closed. So when the atria is pumping blood into the ventricles, this valve is closed. And now when the ventricle contracts, this valve opens and you're pumping blood. Okay, if the valve is healthy and functioning, it opens like that, fully. Okay, and the blood is pumped through the aorta. And remember, it's moving at about a foot a second at that point. Now, if this valve is unhealthy, if it's like that, so stenosis of the aortic valve, you have leakage, and if it doesn't open properly, now it's opening narrowly, so it hasn't opened properly. So if you have a narrow valve, what happens? The blood is going faster out there. Okay. When the blood is going faster, what does that mean? The pressure is lower. Okay. And that's how they sense if your valve is healthy. Okay. Clinician, okay, so if the aortic valve through which the blood exits the left ventricle becomes narrowed, a condition called aortic stenosis, it can lead to significant health problems. One of the diagnostic criteria is a drop in pressure, the blood traverses the wall. Okay, if it goes faster, the pressure is lower than normal. And if the pressure drop exceeds 40 millimeters of mercury, a diagnosis of severe aortic stenosis is warranted. Remember, what's your blood pressure? Normal head blood pressure is 120 by 80. Okay, so when the left ventricle pumps, the pressure here is 120 millimeters of mercury. Now, if you have a valve like this, the blood is flowing faster, pressure drops, and so if the high becomes 80, then you have a problem. Okay? So that's what, so if you do a sonography program, you're measuring blood flow velocity and all that kind of stuff. Okay? So clinicians use Doppler echocardiography to measure the speed of the blood upstream and downstream of the valve. Okay? The speed can then be used to compute a corresponding pressure drop. So in one patient at the left ventricle, if the left ventricle is ejecting the blood, measurement shows a speed of 1.1 meters per second upstream of the valve. Okay, so remember the normal flow was about 0.3 meters per second. Yeah. And 3.5 meters just past the valve. Okay, so this is the speed before the valve, and this is the speed just past the valve. Does the patient meet the criteria for stenosis? Okay, so what you have done is you measured the blood flow velocity here and velocity there using echocardiogram. It's 1.1 meters per second here and 3.5 meters per second there. Okay, so we're going to use Bernoulli's equation to calculate the pressure drop. Okay, so if it's going too fast here, the pressure drop, the pressure here minus the pressure here is what's calculated. Okay, and the pressure drop, so here are the speeds, here is the density of blood, and the pressure drop is 44 millimeters of mercury. Okay, so you want a pressure drop of less than 40 millimeters of mercury. Okay, so if it's greater than 40 millimeters of mercury, that means you've had stenosis and blood is going too much faster, and the pressure drop is higher. Okay, so to check if your valve is good, what you're doing is you're measuring the speed of blood here and here, before and after the valve. If the valve narrows, then the blood speeds up too much. And that's how you detect. Okay. And now you'd have to have valve replacement surgery. Okay. All just from measuring the speed of blood. Okay. So that's what this problem says. All right, I guess we'll stop here today. Okay.